the lab one, the amount of colonies depends on how many dilution used. Based on this experiment, the dilution has been used about 10 negative 2 and 10 negative 3. When the amount of the dilution increase, the number of the colonies are increased. The type of agar also depends on the type of microorganism that grow on the plate. The plate count agar PCA are used to detect the bacteria. Contain agar for coliform such as Salmonella, Shigella, coliform bacteria. And last but not least, PCBS agar are used to detect the Vibrio species.
Okay, for McConkey agar, its appearance of colony is colorless and translucent, mean the microorganism that grow on the plate are Salmonella, Shigella, and others.
For TCBS agar, thiosulfate citrate sucrose agar. From the observation, appearance of colony is yellow colony. So the Vibrio species that grew on the plate is Vibrio cholera. Okay.
Okay, for discussion, lab 2, antibiotic analysis, residue. Based on the result, the visible transparent zone was formed around antibiotic disc, which is ampicillin, penicillin, neomycin, and moxicillin. The sample was positive with a zone size range from 0.3 to 10 millimeter, and then neomycin residue is flash fish in flash fish sample were dominate followed by penicillin, amoxicillin, and ampicillin. So there was low chemical hazard in flesh fish and it's safe to eat. Only a small proportion of animal dry foods are consumed raw, so the cooking process is very important for the reduction of antibiotic residue in food. For question 1, in the lab manual, the disadvantages of using vegetative cell compared to spore are higher susceptibility to radiation than for spore and vegetative cell non-refractive appearance under microscope.
The common antibiotics used for aquaculture practice is tetracycline, which is mainly in hatchery, for prophylaxis in fingerlings.
For the Q3, based on the experiment, we can see experiment 2 in screening of seafood sample for the presence of antibiotic residues using one plate. Test modified from Cytonu, Armosin 1994 has better and more reliable result because the diameter zone of inhibition on the agar appearance more than in experiment 1. Screening of seafood sample for the presence of antibiotic residues using this diffusion method. Reference modified from Calbelli and her friend in 2008 and Baka and his friend in 2010.
Question 4. Other than Bacillus spp, name one species of bacteria that used to screening antibiotic residues are Pseudomonas aeruginosa and Staphylococcus spp because it contributes to high tolerance to antibiotic treatment and the recalcitrant nature of infection due to the permeability barrier afforded by its gram-negative outer membrane.
For the question 5, experiment yield positive results. The most appropriate action that could take in order to confirm the test are repeating the experiment using the same procedure and conditions to get the accurate result. Other than that, communicating and presenting the result to others.
Okay, this is discussion for laboratory 3, parasite inspection. Uh, for the result, we can see that there are 7 parasites that can be found and easily traceable. Usually, parasite can bring disease and death to the host. Example of parasites are nematodes, which is roundworm, trematodes, fluke, and testodes, which is flatworm. Next.
Okay, for clinical sign of fish and human with parasite infection, in fish at external body, we can see the cloud eyes or pop eyes or white patch on the body or redness. In area internal body, warm inside of the body of fish. And the symptom of fish, they will lose appetite or erratic swimming or gasping for air. And for human symptom, they will fever or diarrhea or nausea and tingling.
Common disease caused by parasite. First, fish ichthyophonus, nanophytus, salmonella, and whirling diseases. For human, anisakhiasis and diphyllobothriasis and natostomiasis.
Okay, next, for the impact on the aquaculture, it can cause loss of production, increase cost for treatment, increase time consuming, and after that, it also can affect the human by eat raw fish and undercooked fish.
How to prevent parasite infection on fish? First, avoid disease outbreak. Maintain water quality or reduce stress on fish. Provide the nutritious diet for the fish. Second, avoid overcrowding or overstocking